Okay, good afternoon, everyone. It's my pleasure to introduce today's Science of SLAC lecture series speaker, Dr. Hassan DeMirci. Hassan is an associate staff scientist at the Pulse Institute. He received his PhD from Brown University in 2007. His thesis project actually received the Protein Data Bank Award from the American Crystallographic Association. He continued at Brown as a postdoctoral researcher working on bacterial ribosome genetics. In his lecture today, Hassan will describe what is known about ribosomes' complex dance in decoding DNA and RNA instructions and translating them into the language of proteins. And he will describe new research at SLAC that seeks to unveil the still mysterious steps in ribosome function. His lecture is titled Dancing Ribosomes, The Dynamics of Decoding. Please join me in welcoming Hassan.
Thank you. Can you hear me? Thanks, David, for the kind introduction, and thank you all for coming here today to listen to my talk. Dancing Ribosomes and Dynamics of Decoding. This was the cover, the Dancing Ribosomes, to attract more people, to tell about a horrible story. So, what is ribosome? Does anybody know what is ribosome? What it does? Okay. So ribosome is this tiny machine in the cell which slowly translates the genetic information that we have in our DNA slowly into the three-dimensional form that you see, basically flesh and blood. And decoding means deciphering and decoding that genetic information. I was going to tell about this beautifully choreographed natural dance, a beauty of nature. But we can also use the research that we're conducting here about ribosomes to tackle a really major problem that we're facing today, the antibiotic resistance. Here's the outline of my talk. First, I'm going to introduce infectious disease. I cannot talk about all the infectious diseases, so I'm going to pick one. And I'm going to talk about the translation, the protein synthesis, and talk about the technique that we use to study the ribosome and ribosome complexes. And at the end, I'm going to tell you a fascinating story about a small drug, streptomycin, which binds to the bacterial ribosome and inhibits the ribosome function and kills the bacteria. And at the end, I'm going to tell about the future of what brought me here, what type of research that we can carry at SLAC. You will see this kind of articles more and more. This was just from New York Times, I think yesterday. Yeah. So the rise of antibiotic resistance. It sounds like rise of machines, the Terminator, like some apocalyptic future, right? That's pretty much it. I mean, they here talk about the report of the WHO, and the antimicrobial resistance is a serious problem. It's so serious that it threatens the achievements of modern medicine. Yes, modern medicine can cure cancer, can help you organ transplant, but the antibiotic resistance can pretty much eliminate all the gains of the modern medicine. And whatever we can do right now, a simple bacterial infection can swipe away all the benefits of the modern medicine. And they also say that the post-antibiotic era in which common infections and minor injuries can kill, far from being an apocalyptic fantasy, instead a very real possibility for the 21st century. So this is real. And the problem is that antibiotic resistance, let me put it this way. If you take antistatin, Lipitor, this drug is going to work as good as it works today, 20 years from now. So there's no problem. If you invest $1 billion to design a drug which is going to work in 20 years, it's a great investment for a pharma company. But the unfortunate event is that antibiotics, if you invest $1 billion, develop an antibiotic, that's not going to work a week from now. There's going to be a resistance. So all of your investment is going to be in the garbage can. So it's not a good investment for a pharma company where they have a lot of stakeholders. And there is more loud alarms coming from CDC and about the nightmare superbugs. Drying pipeline of antibiotics due to lack of R&D in the antibiotic field. I would like to ask this question. What can we do after a superbug infection? A bug which is resistant to all antibiotics. Who wants to answer this question? I would say just pray. There is nothing else we can do. Because modern medicine doesn't work, right? I mean, that's the pipeline is dried and no more antibiotics coming behind. So that's the end of the world. So here I'm showing a few people. On the top row you see the George Orwell, Emily Bronte, and Erwin Schrödinger. They have relatively short lifespans. And here you see the Ringo Starr, a drummer of Beatles. You may think that he's alive, right? And you're right. He's alive and he's partying every day. These people died from a disease called tuberculosis, and Ringo survived of tuberculosis for a reason, and I want him to tell his story and not myself. I was driving to SLAC a few months ago, listening my favorite morning show called Fresh Air, hosted by Terry Gross, and here Ringo telling his story to Terry.
Oh, God.
And I also put the transcript.
Lucky to be here, Terry.
Yeah. Well, then you got sick again when you were 13.
I know. Well,
Tuberculosis, was it?
yeah, but that was from the area I lived in.
Industrial stuff?
Yeah, where I lived, like, not every other home, but it was like, 6 or 7 cases in every street where people were just in the living room dying of TB because they didn't have a cure, of course. And again, God, shone his light on me in 1953 or 54 when they discovered streptomycin. And that's what saved me. So they shipped me off to a greenhouse in the country.
A greenhouse? That's like a
Well,
sanitarium?
Yeah, just a huge greenhouse. But instead of flowers, they put all his kids in there and let us breathe some decent air for a change and gave us streptomycin. And a year later, I came out of there.
So how did you keep busy while you were sick? Had music entered?
So it was streptomycin. I gave you a little bit of fresh air, and if you take streptomycin, you'll be all immune to tuberculosis. So I only gave one story of many of the million people who's infected. These are numbers from, again, WHO. In 2011, 8.7 million people was sick with TB, and 1.1 million among these were also HIV positive. And again, 2011, 1.5 million people died from TB, and almost 500,000 was HIV positive. Why HIV is important? Because when you have HIV, that means you have immunodeficiency, and you need antibiotics to survive when your immune system is not working. And that's why these people are more susceptible to bacterial infection. And these are the first-line antibiotics that are clinically in use today. And you can see that the last one is maybe introduced 40 years ago. And the most recent one is introduced in 2012. There's 40 years of lag in the antibiotic development. It's a very slow process, very costly. So again, I want to use this chance and opportunity to tell that, pharma companies, the big ones, they're all out of the antibiotic research field. So there's no big pharma left in the field. So that's the bad news. But the good news is that there are still individual labs pursuing this antibiotic research and resistance. And I hope that in the future we can collaborate, government, maybe big pharma companies, and academics. So we have to pursue. Otherwise, if we don't, the future is not bright. Like I said, a simple infection. This is a cover from a Le Petit journal from 1912. At that time, cholera means death, right? Similarly, in the future, if you don't do anything, then any simple bacterial infection is going to be basically death. So how we can fight this important problem? Here is the central dogma of biology. What it really shows is that the flow of genetic information. DNA makes messenger RNA, makes protein, makes us. So I ask, what is ribosome? Ribosome is catalyzing the last step here, which is arguably the most complex step of the central dogma. Takes the messenger RNA message, which is 4 nucleotide alphabet, and translates into 20-letter amino acid language of the proteins. So DNA replication and transcription processes are very fast and accurate, but this translation process is very error-prone. It's like me. Ribosome does similarly what I'm doing right now. I think in Turkish and speak in English, and I make a lot of mistakes. That's exactly what ribosome does. Thinks in nucleotide language and speaks in protein language, and makes more mistakes than the other two. Here's the structure of a DNA. It's a double-stranded helix. It's like a zipper, and it has only 4 nucleotides in its structure. When you open that zipper, if you see an A, you put a T. If you see a G, you put a C. It's very simple. And the error rate is almost negligible, 1 in 10 million or 10 billion, 100 million. Unlike DNA, which is a very mundane structure, RNA has personality. What I mean by that, it has single-stranded structure, and it can really acquire really complicated and sophisticated folds, and not just carrying the information, but it can also serve as a catalytic molecule or structural, like in the case of ribosome. Actually, ribosome has 2/3 of RNA in its structure. And here's a more realistic 3D structure of the RNA. And ribosome takes the language and translates into a different language of the protein. The protein structure has basically 3 elements. Alpha helices, which looks like a rotini pasta. There's these small arrows, which we call them beta strand. And there are loops connecting those secondary structures. Depending on the sequence of the DNA or messenger RNA, this could be a big protein with shuffled secondary structures. So genetic information dictates the structure of the protein. So this is an artistic depiction of the ribosome translating the message and making the protein. It has 2 subunits, large subunit and a small one. They come together, and you can see this very clearly in this 80-feet sculpture. It's kind of, we can see with our eye. When they come on the messenger RNA, they start dancing. While they're dancing, they produce this, I think it's very inaccurate depiction. Usually protein is not fold over here, but it comes from the bottom of the 50S, but it's okay. So we can see this by naked eye, but the problem is that how we're gonna see these tiny machines within the cell, which is like only maybe like 200 angstrom to 200 angstrom to 500 angstrom. We need a lens. What if I tell you that there is no such physical lens that we can look directly to the cell and see how ribosome works? Instead, we're using a virtual pseudolens called X-ray crystallography, and I'm going to talk about later. Let's assume that we can see ribosomes. How do they look like? It looks like this wet noodle soup. That's how I describe. This is a magnificent machinery. There are many labs all around the world, spend decades of work trying to understand how this machine works, and we still barely scratch the surface. It's so amazing. All of these wires somehow interconnected, and they communicate like from all the way from one end to the other. So here is the small subunit, which I described as a brain of the ribosome. And here is the large subunit, which is, again, I'm going to describe it as the hands of the ribosome. So while small subunit is deciphering the genetic information, the large subunit kind of connects the small building blocks of the proteins and synthesize the whole proteins. So now we see how ribosome looks like, and I'm sure you understand how it works, right? Okay. Now I'm going to split this ribosome like a clam, and we're going to look at it from the subunit interface. So here is the crown view of the large subunit, and this is called the chick view of the small subunit. You can see the beak, the body, spur, and the head. The transfer RNAs, which brings the building blocks of the protein to the ribosome, they enter from the acceptor site. They move to the peptidyl transfer site, where they catalyze the peptide bond formation, and they exit the ribosome from the E site. So, as I promised, I'm going to show this beautiful choreographed dance of the ribosome. And by the way, there's no such association or no such website. And here is the dancing ribosomes. This is the small subunit, the brain. This is the messenger RNA, which is transcribed from the DNA. These are 3 watchdogs making sure that the first transfer RNA, which is carrying the first amino acid, align with the message with a perfect complementarity. And after they align the first tRNA, the watchdogs live, the small subunit joins with the large one, forms the ribosome, and now the last one is going to be released, and ribosome is ready for the elongation. So this is the tRNA carrying the first amino acid. The second tRNA is delivered by a factor called EFTU, and you will see a burst of energy, and the second tRNA is going to be aligned with the first tRNA. Now they're going to form the peptide bond right over here. So this is the second step. Now we have to translate the message on the ribosome. Again, we need another factor called EFG. Again, we'll see a burst of energy. That's the driving force for the translocation. And ribosome is ready for accepting the third tRNA. Now we're going to see a fast forward and bunch of tRNAs coming and elongating the nascent polypeptide. So now it's exiting. So tRNAs coming from the A site, moving to the P site, exiting from the E site. And it happens 20 times per second. And while they're doing that, they're forming the peptide bond in the large subunit, and it's kind of exiting the ribosome through a tunnel called exit tunnel. And this is the protein folding on the ribosome. After a while, there's a message, instead of coding for an amino acid, coding for a stop, and this is a molecular seizure called release factor. It clips the nascent polypeptide and release the protein, and we recycle the two subunits by a factor called recycling factor and a second molecule of EFG. After the burst of energy, it's like a wedge, splits the two subunits apart, and it's ready for another round of translation. So now we see this beautifully choreographed dance of ribosome, but is it really that beautiful and perfect? So this is a scene from one of my favorite movies called Scent of a Woman from 1992. Al Pacino here is enacting an old, completely blind army veteran. They go to a restaurant with his friend Charlie, and they meet with Gabriela Anwar there, and he wants to dance with her. And he's asking, would you like to learn tango, darling? And she says, right now. A blind man is asking you to tango with him. What would you say? And he says, I'm offering you my services free of charge. What do you say? And she says, I think I'd be a little afraid. And he says, of what? Afraid of making a mistake. And he says, no mistakes in tango, not like life. It is simple. That's what makes the tango so great. If you make a mistake, get all tangled up, just tango on. That's exactly what ribosome does. Ribosome makes mistakes, like I do right now. I'm making a lot of mistakes, but I keep tangoing on, right? That's what ribosome is doing, exactly. If it becomes very accurate, if you make the proteins faster, then you're putting more error. If you make it very slow, then life is gonna stand still. There's not gonna be life. So putting error while making proteins, it's okay. We're doing the same thing while we're replicating our DNA. We're putting a little bit of error. So that's the drama in our life. That's the evolution. If you don't make mistakes, then life is going to be without any drama. So what kind of model system that we work with? So here is the Grand Prismatic Spring of Yellowstone. This beautiful colors that you see here, the rainbow, is painted by a bacteria called Thermus aquaticus, and I work with a close relative of this bug called Thermus thermophilus. Why do we work with Thermus thermophilus? In year 2000, our lab switched from E. coli, one of the workhorse of biology, to Thermus because the crystal structures of the ribosome from Thermus started emerging, and also this bug is non-infectious. So when you go to shower, take a hot tub, literally, you're covering your body with Thermus thermophilus. And even though you feel clean, you're covered with bugs. So, and if you dump this Thermus thermophilus to the sink, below 55 degrees Celsius, it's basically not alive. It cannot grow. So it's biologically safe to dispose it. The best part is complete competency for transforming. So the bottleneck in genetics or genetic engineering is giving the modified DNA to the organism that you want to modify. But Thermus thermophilus is like a vacuum. So you just feed them with the modified DNA, it just sucks it naturally, and then you modified it. So it's just so great. And we know the complete sequence of the thermus, so there's no surprises. We know exactly what we work with. The best part for me, it is sensitive to most ribosome targeting antibiotics. What if I tell you that half of the known antibiotics today on the market is targeting ribosome? And this bug is sensitive to all of them. So it's a great model system. It's even better than E. coli, the workhorse of biology. So now I want to talk about the technique that we use, the pseudo-lens, the virtual lens that we use to look at the ribosomes in the cell, called X-ray crystallography. X-ray is discovered by Röntgen, and he received the first physics Nobel Prize in 1933. And this is one of the first X-ray images taken by him. I think it's the left hand of his wife, and this is their wedding ring. It's a very famous photo. Why do we use X-rays? Here's the light spectrum. So human eye can see anything beyond 40 nanometer in length. So if you go something smaller than 400 nanometer, for example, if you want to see the atoms or molecules, proteins, viruses, you have to use X-rays because their wavelengths are helping us to see those short distances. So we generate X-rays by using an electron gun, and then we hit the electrons to a metal target, and then that generates the X-rays. We collimate the X-rays, make them parallel, and generate crystals of the biological systems, and hit with the X-rays and collect the diffraction pattern onto a detector. Why do we use crystals and not just single particles? Because X-rays are very poor diffractors of the biological systems. And typically, biological systems consist of carbon, nitrogen, sulfur is the biggest molecule. So they don't have many electrons, and that's why they don't really diffract the X-rays as good as the heavy atoms. How do we get the crystals? So we grow bacteria, and then after we grow them, we break them open by using high-frequency sonic waves, sound waves. And then after we burst them, we use very robust computer-assisted purification systems. And once we purify ribosomes or any proteins that we want to work with, we just mix them with different, I would say like even table salt or table sugar is something you can use for crystallography. And then we get the crystals in these plastic plates and then collect data and solve the structures. So crystals come in different flavors, different geometries. Colors are kind of deceiving. This is a polarizing microscope, so they have no color. And here's more crystal photos. They look perfect. Sometimes they look perfect, but indeed, they're not that perfect. Again, I want to make a comparison, how the human eye can see and how we see with the X-rays. So here is a plaster model of a ribosome. So it's a plastic model, and the incident light reflected through the surface, and the human eye can focus the reflected light on the human retina. That's how we see. But there is no lens that can focus the X-rays. So here I show this cube, which is a crystal. It's the wrong depiction, only showing one ribosome in it. But imagine there are a million copies of the ribosome in that crystal. And once the X-rays hit the crystal, they kind of, the diffraction pattern collected on a 2D detector. So basically, diffraction pattern is Fourier transform onto a flat surface. So you took a three-dimensional object and you collected data in two dimensions. So you lost that volume, the 3 dimension. And then we use some of the prior knowledge about the object that we study and some algorithms, and we reconstruct the original object that was in the crystal. Who wants to see what's in the box and what X-ray crystallography looks like? Okay, I'll give a split-second view. Okay, this is how it looks like. So we back Fourier transform the diffraction pattern and reconstruct this beautiful electron densities. I would call it my love of life, but my wife is going to hate me. So these are electron densities, And this is my passion. I love looking at these electron densities. Remember that I said we have some prior knowledge. And you see some continuous parts and some extra pieces sticking out. First, we built the main chains of the protein. And these are kind of sequence that we extract from the DNA. And then, remember that ribosome was synthesizing proteins? That exactly every blue bond that you see over here is put together by ribosome. And once we built the main chain, there are extra pieces sticking out. Those are the side chains. Again, gives the flavor to each protein. So each side chain has different chemical properties and determines what that protein is going to do, what kind of surface it's going to expose when it's in the cell. So now you know my technique, and you know the model system that I work with. And the major question, the main question that I want to answer is, what do the antibiotics and mutations in ribosome tell us about decoding? Again, I want to tell about what is decoding, right? If you don't know what is decoding, how I can study it. So here is the bacterial genetic code. Remember, DNA has the code, and ribosome translates it. Every 3 nucleotide that you see here codes for an amino acid, each triplet called code. There are 3 exceptions. Those are the ones which is signal for a stop. Remember the movie, there was a molecular Caesar coming and clipping the protein? Those are the signal for that Caesars that comes and clips the protein. There are certain peculiarities about the genetic code. It doesn't really matter what's the first and second nucleotide is. It always codes for the same amino acid. Again, the third position doesn't really matter, but the first and second position has to be super accurate. Then the question is how ribosome knows what's in the first, what's in the second, and what's in the third position. So we're going to look at the details of this decoding. It's a very fast process. Like I said, almost 20 amino acid formed linked together on the ribosome, and ribosome makes only 1 mistake in 10 peptide, 1,000 to 10,000 peptide bond formation. This is the messenger RNA, and ribosome puts the messenger RNA together with the transfer RNA, which is the adapter molecule. And biology is all about proximity. If you put 2 things next to each other, then the life goes on. And ribosome aligns these 2 molecules, the messenger RNA and the transfer RNA, together at the decoding center. And the first and second position of the codon is kind of strictly monitored by the ribosome, but if there's a mismatch in the third position, this amino acid, which is attached to the 3' end of the transfer RNA, is accepted as the correct amino acid and correct tRNA. This is called cognate tRNA. If the same mistake, GU mistake, in the first or second position, ribosome can discriminate these 2 tRNAs very effectively and eject it from the ribosome, saying that you're wrong, and the next one comes. So this is called tRNA selection, the decoding. Two-step kinetic proofreading model of the decoding. How simple is this slide? I'm going to make it even more simple. So now we're looking at a 100-meter hurdle Olympic race. So every step that you see here is a hurdle. But if you are the correct tRNA, the hurdles are so short, maybe a few inches tall. But if you are the wrong tRNA, each hurdle is a great China wall. So if you are correct tRNA, which is in this case the second position, can very easily run all the hurdles and win the game. It's just like a piece of cake. It's a very easy game for her to win the race. She's such a great athlete. She can run this 100-meter race 20 times a second, and she loses only once in 1,000. So I think it's great stats. So now we're going to look at it in atomic detail. I mean, this was just the overview. So now we're going to focus in and look at how ribosome discriminates between correct and incorrect tRNAs. So the first step is the binding of the tRNA. So here's the messenger RNA, position 1, 2, and 3. And ribosome, remember, aligns the transfer RNA with the messenger RNA at the decoding center. And now, ribosome is going to use a very elegant, a very nice way of monitoring this binding over here by using these two fingers. So the gray was the unbound state. Once the correct tRNA is bound, ribosome changes the conformation of its decoding center. I would call these two residues the brain of the ribosome. So what it monitors is that the isosteric nature of this binding here. What is isosteric? Iso means same, steric means geometry. So there are 4 nucleotides, A, U, C, and G. A and G is a little bit larger than U and C. So if you put A and G together and U and C together, it's either too short or too long, right? But if you flip the other way, A and U, G and C, when you do this, you're only measuring the distance. So it's independent of the sequence. If the geometry is correct, ribosome doesn't even care about the base pairing. It's just measuring the distance between the one end to the other. So these two fingers flip and fits like a nice glove. If this mismatch over here is a little bit bulged, a little bit bigger, then it's the wrong tRNA and it's easily rejecting the wrong tRNA from the ribosome. So that brings us to the second step over here. There's a activation step. So this was the recognition, and the first and second position is monitored by these two fingers. If you remember, the third position was very relaxed, right? Doesn't matter really what's in the third position, it always codes for the same amino acid. Because this residue over here comes with an angle, not with the straight flipping. So that gives the relaxed recognition in the third position. So now I'm going to put it back. So we're going to look at the whole thing in the context of the ribosome. So this is the brain part, and here is the tRNA analog. So we're going to cut the head of the ribosome over this plane, and we're going to look at it from the top. So ribosome goes from an open to a closed transition during the decoding. So if there is a correct tRNA, ribosome kind of clinches the tRNA in the decoding center and stabilizes it in the ribosome. If there is no correct tRNA, there is no closing. So it only closes when there is correct tRNA. There are also mutations in the ribosome, so this dynamic is kind of optimized in a way that makes ribosome maybe one mistake in 1,000 to 10,000. But we can make mutations over here or over here. For example, mutations here is favoring the closed state, make ribosome easily close even if you have wrong tRNA, and makes the ribosome even more error prone. On the other hand, if you make mutations over here, it favors the open state. Now you're started rejecting even the correct tRNA. That makes ribosome very accurate. So this version of ribosome, me speaking 100 times slower. So would you be willing to stay here for another 10 hours to let me finish my work, or do you just want me to make more mistakes and continue? So this is like hyper-accurate ribosome phenotype. And these bugs grow very slow, and the whole media is overtaken by the natural bug. So that motion was, we were looking from the top, and it's usually the orange color is closing towards the green helix that you see. So now I'm going to put the full-length A-site tRNA, P-site tRNA, and E-site tRNA. These tRNAs delivered by a factor called EFTU, and the recognition over here induces the activation of this factor. So imagine that shoulder or the body is closing towards the factor. If this is the factor, I'm kind of reaching to the factor and pressing a button saying that, okay, we have the correct tRNA, and that activates this factor over here. That's the step, that's the irreversible step. So up to here, everything is reversible, but once you have that domain closure and activation, then you're in one-way street. So either you're going to be, again, proofread at some point if you're the wrong tRNA, or you're going to go ahead and form the peptide bond. So now we have the activation. I'm going to put the whole ribosome together. So after the activation, the factor released. So if you are the correct tRNA in the A site, there is this domain closure that kind of stabilizes the tRNA in the active site. And there's remarkable torsional strength. It wants to go this way. But if you're the wrong tRNA, there's no domain closure, and kind of you're loosely bound. And at this stage, if you are here by mistake, you will be gone. So that's called proofreading. But if you're the correct tRNA, there's domain closure, and you swing, and you form the peptide bond. Shazam. That's the biology. So that's how ribosome catalyzes peptide bond. Again, I want to ask the same question. What do antibiotics and mutations in ribosome tell us about the decoding? So this is a quote from a 40-year-old book called Molecular Basis of Antibiotic Action. It's written by towering figures in the antibiotic field, and this book is still as good as it's written 40 years ago. If you read it, it's absolutely up to date. I mean, there are slight advancements, but we're still where we are maybe like 40 years ago. There's not much advancement in the antibiotic field. So they say that the diversity of effects produced by this aminoglycoside and the ingenuity with which they have been pursued have made the streptomycin story the most fascinating and complicated one in the field of anti-ribosomal action. So this is said by towering figures, and it guarantees that I'm going to tell a fascinating story. This is a classic paper by Julian Davies et al., and in this paper they showed that ribosome has a decoding center that deciphers the genetic information, and the drug streptomycin increases the error rates. So this paper shows that the streptomycin is targeting the ribosome, and that's how it's killing the bacteria. So this is a paper that we published a couple years ago, and we showed that actually this was a repeat of another study, but we add extra layers. Streptomycin binds, that's where near the heart of the decoding center. And it distorts the decoding center, which I'm going to show in the next slide. So I have to explain a concept called superposition. So if you have two structures, one of them is empty and the other one is bound with the drug streptomycin. So what we do here is that basically just superimpose them together, and if there's any difference, you can see them. And here, if you see any misalignment, typically what you see over here, that's where the differences are introduced by the drug. And streptomycin literally distorts the brain of the ribosome. So those two residues, those two critical residues that you see over here, is flipping and monitoring the first and second position of the code. So if there's any misswing during that decoding, you're not really reading the message, but you're just, instead of translating Turkish to English, if you give me streptomycin, I'll be speaking maybe Chinese or maybe some unrelated language. So that's exactly what streptomycin does. So this is the movie that I made, and that shows like how the drug distorts the ribosome. As I described before, this whole thing is a live machine. So if you do like mutations somewhere, then you see the whole thing is moving. Sometimes there are connections in the whole structure that talks to each other. There's a signal relay. Drug binds right here, and that distorts one helix, but then that distortion over here is kind of propagated all over the structure. So it's hard to chase how these signals are relayed from one part of the ribosome to the other part of the ribosome. And this is kind of the brain. Remember those two residues flipping 20 times a second? Now you lose that support, which is holding the flipping residues. You disengage, you kind of become more, I would say like more marshmallowy, more softer. You lose your rigidity. So if I summarize, drug streptomycin distorts the decoding center, and that's how it kills the bacteria. But the mutations in ribosome gives resistance to the drug streptomycin. So first I'm going to talk about the resistance, and I'm going to talk about an extreme case of resistance. Instead of being resistant to the drug, the bacteria becomes dependent on the drug. So I'll talk about those junky ribosomes after I talk about the resistance. So this is a paper accepted 2 days ago, so it's as fresh as it gets. And what we show here is that there is a central part of ribosome. There is this 3 residues come together, the gray part. All the colorful stuff is the mutation. So if you change any of these residues, one or the other, it rearranges the structure. But what you see is that the backbone, the overall structure, is not really distorted too much. And here where you see the drug binds. So you don't really see much difference in the overall structure. We also solved the same mutant ribosomes with the bound drug, and we showed that the mutations still, allows the ribosome to bind to the drug streptomycin. And we repeated the same experiment for a bunch of other mutations. Here, for example, a mutation in residue 914, which is right here. It changes the nearby structure, but again, like the overall structure doesn't really change much. So the resistance phenotype is a very tiny changes in the overall structure. And when we put the drug, we again determine the binding of the drug with almost like wild type. So this kind of suggests that the mutation doesn't really eliminate the binding of the antibiotic, but somehow the dynamics of the downstream. So it still binds, but since you lose that rigidity, while that signal has to relate to the decoding center, it's kind of, instead of hitting a rock, you're hitting a cotton. So it doesn't really propagate that message. So that's kind of, what we're seeing with the resistance. And if I summarize, the structural data that we obtain from these mutations suggests that the tertiary structure of the ribosomal subunit is very robust to mutations. So it's a 2.5 megadalton structure. So this allows the bacteria to become really robust. So if you give them a drug, it just picks up a few mutations and keep going. So it doesn't really kill it. And the mutations has always a fitness cost. Maybe it slows down or maybe little bit, not too much, but keep you going. So now I'm going to talk about the junky ribosome. Now we have a mutation. I think it's D mutation, which makes you resistant to the drug streptomycin if you're a bug. But when we take the drug off of the media, then the bacteria dies. So without knowing, the bacteria become dependent on the drug streptomycin. So you can see that this is the crystal morphology that we see with the wild type, the functional bacterial ribosome. If you have a point mutation in anywhere in that giant structure, you get this new crystal form. So now we're looking at a dependent state. This structure, which I'm going to show later, is unlike the other ones that I showed before, which was slightly different, right? This is dramatically different. So it's highly distorted structure. Now we need a drug to become functional again. So if you take these ribosomes and give them the drug, or if you have a second mutation elsewhere which suppresses that dependence, you again obtain the other crystal form. So by looking at the crystals, you can say that this one is functional state and the other one is non-functional state. So when we look at the crystal lattice, we can see that in the wild type, these two proteins from the neighboring subunits touches each other. In the mutant, these proteins slides almost 150 angstrom and touches to the other part of the ribosome that you see here. So instead of touching each other, they're touching somewhere else. So crystallography speaking, this is like 10-mile difference between the two structures. It's not really a tiny difference. Now I'm showing again the brain, the decoding center of the ribosome, and this is where that mutation is going to be. This is calculated electron densities of the decoding center of the ribosome, and here's the new model that we built. And now I'm going to get rid of the electron density and show the cartoon. Unlike the other part, which was far away from the decoding center, this mutation is right next to where the brain is. So this slight distortion over here, it kind of puts the ribosome in a state where it doesn't function anymore. But when we give the drug back, that distortion is kind of restored. And in the presence of streptomycin, this mutant ribosome, by retaining the mutation and the antibiotic, can keep going on. So either the antibiotic or the mutation itself is lethal, but when you put them together, they function again. So the streptomycin-dependent mutations result in extreme distortion of the decoding center, and the physical presence of the drug partially restores the deformation of the decoding center. And with that, I would like to change gear and talk about the future research. So this was the past research that I was carrying. And I always show like the snapshots of the biology, right? Before drug, after drug. And I show some movies, which was not scientific, mostly science and fiction. So what we did is we take a snapshot, we take another snapshot, and we interpolate the two structures. And that's where we guess the intermediate states could be. So I came here to SLAC to take the advantage of the LCLS. This is, as you all know, the world's first X-ray free electron laser. And I'm not going to talk about much, but I think the electrons are accelerated in a kilometer tunnel with 16.2 gigawatt of peak power, and they enter this engineering marvel called undulator hole. These are the these are the magnets which are aligned with less than a micron precision. And once the electron exit the undulator hole, they generate X-rays which are 1 billion times brighter than a synchrotron can generate. So we typically carry out our experiments at the CXI end station. So from here, you can see that we have 1 billion times brighter X-rays for a person like me, crystallographer. Future is very bright, unlike the antibiotic field. So we want to put some light to the field of antibiotic research from the LCLS. We're trying best we can. And this is the technique called diffraction before destruction. So typically, LCLS beam is introduced in small packages, short pulses of 50 femtosecond or less. And once the sample is intersected with the X-rays, it explodes via Coulombic explosion. But before that explosion, we collect the diffraction data. So I love giving this example. I got my PhD from Brown, from a tiny state called Rhode Island. So imagine you're driving a Ferrari from Massachusetts. You enter with 300 miles per hour to the Rhode Island, and there's a highway patrol waiting for overspeeding people. By the time when they notice that you're overspeeding in Rhode Island, you're in Connecticut. So you already entered and exit. So that's exactly what happens here. So I came here to work with Mike Bogan, and it's NPI, non-periodic imaging of pulse group. But now instead of NPI, it's called no PI because he left, and we're still here. We're still here. Everybody is in the lab, and we're still doing some cutting-edge stuff. And this is the injector that NPI developed. It uses a very mild electric field to introduce the tiny crystals to the X-ray beam. So we collected some diffraction data from our ribosome crystals, and we showed that they have some potential to diffract, but it was collected with an alternative jet. Now we want to do our experiments by using this jet. But beyond doing this kind of experiments, which is not why I am here, not just collect regular data, we want to mix those tiny crystals with the antibiotics, maybe decoding complexes, or peptidyl transfer intermediates. Like I said, we barely scratched the surface of ribosome field. And most of these stuffs that I talked about, they're still highly disputed topics, despite the fact that there are a lot of Nobel laureates in the field. So what I think is that by using LCLS and taking the advantage of this very brilliant X-ray sources, we can carry out time-resolved X-ray crystallography by short mixing times. And after those mixings, we can collect data at the LCLS. Why LCLS? Why can't we do these experiments at synchrotron? Imagine, in 1 second, there's 20 peptide bond formation. We're talking about millisecond. Ultra-fast science here at SLAC means attoseconds, femtoseconds. But when I talk about milliseconds, I'm a biologist, an X-ray crystallographer, it's ultra-fast. Millisecond is as fast as it gets at biology. So if we can achieve this time resolution at LCLS, it's going to put us in a whole different world. So now we can study things that was impenetrable by synchrotron crystallography. So what I mean by that? So before I showed a drug unbound ribosome, and I showed a drug-bound ribosome. This is Sean White. And what you see here is that, like, first, a very simple rotation, right? That's what we were doing, like just interpolating the two structures that we had before. But is the biology that simple? Like intermediates, are we really capturing any of the important biology? Can we find another intermediate so we can hit that intermediate state by a new generation antibiotic? So we want to explore the possibility of finding those intermediates. And I want to finish with a crescendo. Actually, this is how we how he came from the first state to the second state. And this is what biology is all about. And I believe we can capture these cool biology by using LCLS. This is where the future is, and I think it's going to attract a lot of biologists coming here. And everybody that I talk to, everybody wants to do the experiments, but I think LCLS is star right now. Everybody wants some beam time, so we're still fighting for that precious beam time to carry out these experiments. And with that, I would like to first thank Mike Soltis. He's my new advisor and colleague and collaborator. Jody Puglisi from Stanford campus is my second advisor here, and also kind of, I feel like a adopted child everywhere. So I came here to work with Mike Bogan. He's still giving us good advice, and these are wonderful colleagues, Hartawan, Ray, Claudio, DeMirci, and my PhD and postdoc advisors, Al and Gerwald. Steve was the person who helped us to get those mutations, the mutant ribosomes. I would like to give a very special thank to Venki for letting me, or giving me the wonderful opportunity to visit his lab to learn ribosome crystallography. And it was Anne who showed me how to do the crystallography of ribosomes. And Frank helped us some of the data collection. Right now, I'm supported by the grant from Stanford Institute for Chemical Biology, and previously we funded by NIH, NIGMS. And with that, I would like to thank all of you for being here. And I'll be very happy to entertain if you have any questions.
I have a question about the crystallography. When you do the crystallography on the synchrotron source, I guess you need to align all the ribosome in the same direction to get diffraction. How do you actually align them?
So for the LCLS data, there's no orientation, but there's
For
more.
the previous one, when you have the crystal and you...
But now, like you said, there's no rotation, simple rotation on a goniometer, right? There's random orientation of the crystals. How we merge the data? We use
not for the LCLS, but previously when you did the synchrotron and you had to have several ribosomes in a crystal.
Yes.
So I'm wondering why the ribosomes are aligned in this. The question is, do you need to align them in the same direction to get the diffraction pattern, or not really?
Ribosomes in the crystal?
Yes.
They are aligned. That's why they are crystallized. If they were not aligned, then they wouldn't form that nicely ordered crystals. There are certain parts, certain fraction which is not aligned. They generate the mosaicity problem, but we still, solve the structures because most of them are aligned in one direction.
I see. So it's the crystal of ribosomes. Okay.
Yes.
I guess one question I had
All right.
is, you had these statistics of the ribosomes, that they're actually, forming these links at 22 links per second. They're accurate to a part in 1 to, 1,000 or 10,000.
Yes.
How do you know these numbers, or how are those numbers?
At the beginning of my talk, I said there are many laboratories working on the ribosome field over many decades. And these are the numbers that we get from in vitro studies, bacterial growth experiments. People spend maybe, light years in terms of human time to collect that data. There's a lot of measurements done after we lyse, we purify each component and then do, radioactive assays and a bunch of other assays to get those numbers.
You show that a very small difference in the structure or in the conformation causes a huge effect in the end result.
Yes.
Do you expect, now when you talk about Sean White, do you expect to see anything in between those two changes, or do you think that even much more subtle changes are already causing enough big changes to be interesting to study?
So there are like multiple layers of those conformational. First, you see the flipping of the two residue, and you see neighboring helices engaging, disengaging, and then you also see the whole head is rotating, right? So just separating the small from the big is gonna be a giant step in the field. I'm not even talking about like the other states. Right now, we don't even know. We think we know, but we don't know how the decoding happens. We don't know if that, domain closure really part of the decoding, or is it just, like I said, these are highly disputed topics in the field today. Like if you can separate those two, tiny versus big rotation, that's a giant leap. I mean, I hope, these are like 101 experiments waiting to be done at LCLS.
Hi.
Hello.
I was reading just a few days ago a really fascinating article about a Japanese scientist a couple of years back that had this idea that viruses are able to penetrate the human cell membrane because they change their, and these are his words, not mine, they change their fractal dimension, fractal structure, okay? There is a shape to these viruses.
Yeah.
And they actually are able, I don't know how, but they're actually able to change their structure. And that enables these viruses to quickly change, adapt, and penetrate the human cell membrane and cause disease. So, getting down to the nitty-gritty of what you're doing with ribosomes, have you any thoughts about that? I mean, are you investigating that? Somehow I get the feeling that viruses are able to cause disease because they change their outer structure. Somehow they're able to penetrate, the human cell membrane. This Japanese scientist thought about the fractal dimension of these viruses. And I was wondering if you have any thoughts about that, or even think about that, or whatever.
So it's unrelated, but
Okay.
my wife is also Japanese.
Okay, good.
I love Japanese people. Come to the question of viruses changing their
Yeah.
shape.
Right.
This is biology. I mean, it's very dynamic. Not just viruses.
Right, not just viruses, right.
Exactly. So proteins move, ribosomes move. It's not just the dance of ribosomes, but it's dance of life. But that's just the first step. The viruses just, they have to penetrate the membrane, but once they are in, they have to do many other things. They hijack the ribosomes of humans to make more viruses or virus proteins. So changing the structure of outer, I would say, capsid, I think it's possible, and I don't think it's just out-of-the-box idea. I think it's expected, but there are many layers. So it's just maybe the first step of that choreographed dance.
Okay, thank you. It's just fascinating. When I read this article, it was like, and I knew that, that you were having this,
Yeah.
this
Again,
meeting.
I mentioned in the acknowledgement, now we work with Jody Puglisi, and we do some HIV-related stuff. So we do some virus-related stuff too.
I had a very basic question about ribosomes study in general. I exposed, but maybe it reflects on what you're doing, which is that this seems like a very complicated thing to have evolved and to be effective. I'm wondering if, we have ribosomes. Are there primitive bacteria that you pick up in, the bottom of the sea that have simpler structures that you could study first and understand that, or?
So we have two types of ribosomes ourselves, not just one. One of them is called mitochondrial ribosome. So this is an organelle, which is kind of a compartmentalized part of our cell which is dedicated to produce energy. And that is, I think, evolved from a bacteria. So we pick up a bacteria and kind of, use it as a slave to make the energy. Those ribosomes are very similar to bacterial ribosomes. Again, antibiotics has a lot of side effects. For example, streptomycin and other aminoglycoside class, which is like streptomycin, has very severe side effects. For example, when you take those aminoglycosides, you may start not hearing. And if you think about the human ear has a lot of rod cells, and those are kind of, run by mitochondria. So you're just kind of, killing the mitochondrial ribosomes, and it's causing the deafness. Again, the evolving ribosome, this is like the optimized structure. Like I said, it's making one mistake in 1,000. There's this 2.5 megadalton structure. Ribosome has itself, I gave this example many times, 8,000 amino acids, 50 proteins, and 8,000 amino acids on itself. So what it means, while you're making ribosomes, it's guaranteed that each ribosome has a mutation, a mistake on it. So if you have 20,000 ribosomes in your each cell, none of them are alike. They're all very different, slightly different. At least one amino acid, I can guarantee that they're different from each other.
So I was wondering if there are simpler structures that do people study the mitochondrial ribosomes and they understand them better?
So mitochondria, I would answer this way. You know the movie Inception, dream within dream? So this is like, first you have to penetrate one membrane and purify the cellular ribosome. You have to isolate first, break the cells without breaking the mitochondrial cell, because you have to kind of prevent contamination between two ribosomes, right? First you have to purify organelles, which is a fraction of the cell, and then you have to cut that membrane and purify the other ribosomes, right? So for crystallography, you need a ton of ribosomes. And getting those two membranes and getting those ribosomes purified for crystallography was a big challenge. But now there are many labs focused on cryo-electron microscopy. So there's 60S subunit, the large subunit from mitochondria, and also the small subunit. It just appeared, I think, today or yesterday. So they're emerging, but not by crystallography, but another alternative technique called cryo-EM. So they're simpler, or I can describe not really simple. Ribosome is never going to be simple because this thing is by itself like 2 megadalton, right? So there's no simple ribosome research. It's complicated, however you like it or look at it.
Maybe we have time for one more question. Okay, if not, let's thank Hassan for a fantastic
Thank you so much.